Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care. Hello, everybody, and welcome to another episode of the Broad Eye podcast. My name is Sean Maloney, and I'm here today with my co-host, Dr. Bruno Fernandez. Bruno, welcome.
Thank you, Sean. Pleasure to be here again.
There you go. This is an exciting episode for us today because today we are sitting with Dr. Cynthia Qian. Cynthia, well, you know what? I'm going to ask Cynthia to introduce herself. So first of all, Cynthia, thanks for joining us today.
Great. Thanks, Sean. And it's great to be here with both you and Bruno. It's, as we were saying, it's been a long time since we got to catch up. And this is a great opportunity to discuss some really pertinent topics with the two of you. Great.
So, as you mentioned, it's been a long time since we caught up. It's probably been a decade or so. And a lot has happened in that decade. We've all grown up. But maybe you can just start by giving the audience a brief background of who you are.
Sure. So my name is Cynthia Qian. I'm a Montreal hometown girl with a broad international background in my medical training. So briefly, I was at medical school at McGill University, where I had the pleasure of getting to work and getting to know both of you at the time when I was doing some research in ophthalmology. Then this was followed by a residency training at the University of Montreal. And then I went on for fellowship in the United States, where I did subspecialty training within the specialty of ophthalmology. So I trained in surgical retina, meaning I take care of both adult and children's pathologies or problems of the retina. This one I did at Mass Eye and Ear Infirmary of Harvard Medical School. And then following that, I also did additional fellowship training in inherited retinal diseases at the University of Michigan in Ann Arbor.
All right, Cynthia, that is indeed very impressive. But as every other remarkable person, right, you're certainly humble as well. And you forgot to mention that in 2017, you were chosen as the young physician leader to represent Canada at the World Health Summit in 2017. In 2018, you voted the Young Professional of the Year by the Young Chamber of Commerce in Montreal. And in 2019, if all that wasn't enough, you were elected one of Canada's top 40 under 40. So super impressive resume. We're very honored to have you here. And you're certainly qualified to talk about the topic, which is retinitis pigmentosa. So if you had to explain to a lay person what exactly is retinitis pigmentosa, how that affect the eye, and what kind of symptoms someone affected would present.
Yeah, so retinitis pigmentosa, as you said, is a very broad term. I think a lot of people are familiar with the term, but they don't necessarily know exactly what it means or what it stands for. And I have the pleasure and I have the privilege of actually seeing many of these patients in my clinic, in my day-to-day practice, where I see patients with both pediatric and adult retinal diseases, many of which are inherited or family-born. So retinitis pigmentosa is actually a general term that groups many different rare genetic disorders. So it could be coming from different gene mutations, and ultimately they all lead to the loss of function and the breakdown of specific cells within the retina, which is the light sensing organ or the sheet of cells at the back of the eye that are able to transmit light as visual signals all the way to the back of the brain to the cerebral cortex. So basically there's a loss of function of those specific cells, which leads gradually to the loss of being able to see clear images and ultimately, in some very severe cases, all light completely as well. So, and, I'm actually gonna add a story here. Actually, Sean, I think, it was actually really interesting because, back when I was a medical student, when we were working in the ocular pathology lab, I think you're one of the first people that spoke about having retinitis pigmentosa, and that really, made an impression on me back then, because obviously in day-to-day interactions in our work, that's not something that was discussed. And it was interesting because one day, while we were working on something, it just was in the context, and you mentioned that you had retinitis pigmentosa, and I was really surprised because at the time I didn't know. And it's not something, knowing you initially from the work perspective was something that I had really, thought about. And it was just, a surprise to me when you first talked about it, because it was very natural, and it's not something that I noticed in, our day-to-day interactions or in the work that we did initially. So I think that was something that I definitely remember from our early interactions and talking about retinitis pigmentosa. So definitely that left a mark on me.
That's amazing. Then and back in those days, obviously I could see a little bit better than I do now. We're talking 10 or 15 years ago. It's been quite a while, but I think I've always tried to orient my work around my strengths as much as possible. So you didn't go out with Bruno and I going out for beers too much after work. You were working out. Then if you would have been into those low lit environments, and when I was walking in the wrong directions everywhere and stuff, you might have noticed a little bit more than in the daylight or the incandescent lights of the laboratory. Yeah. So, but no, that's interesting. That's interesting. So, can you talk a little bit about what the current state of affairs are for treatments for retinitis pigmentosa, and then maybe at the same time what you would see in the pipeline specifically for gene therapy for this disease?
Definitely. So I think usually we like to represent things graphically, where, you have retinitis pigmentosa in its in the lifespan of a person, where it evolves from minor visual signs and symptoms to the more severe forms. And at each different stage, there are levels at which different types of treatment modalities are suggested. So for example, the gene therapy, which is really what's exciting, new and up and coming, covers a good majority of the time span from early disease up until moderate to pre-severe diseases, because basically the idea is that you still have enough cells left that are functioning that will benefit from the effect of potentially salvaged or reverse some of the loss from the disease itself by the gene therapy. Once you get to the more advanced levels, you're looking at treatments such as retinal implants, retinoprosthesis, or what has been popularly known as the bionic eye, where you're not necessarily dividing or categorizing the different types of retinitis pigmentosa so clearly, because at the end of it, all the different types of these diseases lead to similar looking end stage disease, and you're basically just trying to provide the person with better light and motion to alleviate or make movement and avoiding obstacles easier. And then in between these sort of early stage and late stage treatments, you also have other new modalities which are less you need to less differentiate between the different mutations. For example, we're talking about stem cell therapy, and then there's also this new field of optogenetics, where you're trying to bypass the sick or non-functional photoreceptors and trying to stimulate the next layer of cells in the retina, which are the ganglion cells, which will then transmit the message all the way back to the brain. So there's really these, you could say for now, like 3 or 4 different stages of different treatments, but it's really gene therapy that has taken up in recent years. And that's really, there's a hustle and bustle of a lot of excitement, a lot of promise, and there have been actual treatment results through that approach.
I was just going to follow that up with just to kind of put it in perspective for people listening. When you're looking at the progression of the disease, so what you're saying is in those early stages, gene therapy is really the go-to, well, I say go-to treatment of choice, or what we're trying to establish as a go-to treatment of choice, despite the fact that it can be complicated, given that there's a lot of different underlying mutations that could lead to the disease, right? But this is more the stage that we're at when trying to use gene therapy to treat. And then as it progresses, you're losing some of the actual cells, right, that gene therapy would be used to treat some of these photoreceptor cells. And that's where you're saying stem cells or optogenetics comes in. And then and then kind of the late stage disease, you're looking at some, because you don't even have maybe the architecture there anymore, right? Mm-hmm. Like even for stem cells, you need to have some architecture, I guess, for the cells to grab onto, right?
Yeah, definitely. Exactly. And I think that's what is important for, for I think your listeners and for people out there who are diagnosed with inherited retinal diseases to know that it's important to, first of all, to, the earlier the diagnosis, once symptoms start, the better, because then it gives us more time and more options to work with. I think time is retina. So as time goes by, the natural procession of the disease is that there's going to be gradually more and more cell loss. So, the earlier we know what the disease is, the earlier we know what the genetic mutation is, we have much more options open to us in which way to best, tackle the issue and preserve more vision and more functional visual abilities.
All right. So you think about gene therapy, right, which is as I understand, like the treatment of choice, like for patients that are early in the course of retinitis pigmentosa, there must be like a ton of questions on their part on what exactly does it entail to be enrolled in a clinical trial. So what are the usual questions like that they have for you? Like, I mean, the risks, like, I mean, how likely they are to get their vision back, so.
Yeah, for sure. So I think, the first part that's really important for me, I think, is to clarify what this treatment is for many people who have visual problems. I think, for example, when the first officially approved gene therapy for eye disease was approved just last October, a lot of people with vision loss were very excited by this news. But, in general media, it's not very clear that we're specifically targeting inherited retinal diseases. So a lot of people were full of hope. People who've had, for example, horrendous accidents, had retinal detachments many years ago. They come back 40 years later and are hopeful that this is a treatment for them. Because gene therapy, stem cells, these are terms we hear very, very often, right? But we have to kind of give the patient a very clear image of what, first of all, the basic inclusion criteria, for example, to become eligible for it is. So we have to make clear that it has to be people with genetic mutations leading to retinal disease. It, unfortunately, for now, is not for people who have had physical retinal damage, either from trauma or from previous, incidents that have led to surgery. Because, as you can imagine, the eye has to be fully intact physically to be able to accept the insertion of this gene therapy. So an eye that has undergone a lot of change or scar tissue would not be able to tolerate, will reject this treatment. So it's really for people whose eyes, architecturally from the outside, everything is intact. The retina is in place, but it's really at the cellular level that the cells are not functioning fully, or they're losing their function over time due to mutations in the gene coding that was really, a genetic signature that has been pre-programmed since birth. Either this visual loss can come on later in life, during childhood, or even later in adult life, but they have to have at least those two basic key factors. So physically, the eye has to be healthy and can tolerate a surgical procedure. And secondly, they need to have a confirmed test showing that they have the genetic mutation in question that has been shown to be able to be treated through gene therapy.
Let me just follow up on that. So for a patient that go through all that and they do the genetic testing and they show like the mutation that can be treated, so it's a treatment that is approved already, right? So it's not a clinical trial anymore. It's like established treatment like no other. All right.
Exactly. I think that's, like you were saying, that's very important as well. Patients have a lot of questions. Is this, is this testing on me? Is this sort of experimental, science or research? So for the one specific form of retinitis pigmentosa, which is the one caused by the mutation in the RPE65 gene, this is now approved in Canada by Health Canada, approved in all the provinces to be administered as a treatment to everyone who does apply to the criteria or who is eligible. So there should, it's not a trial and error to see which medication is, is usable or whether it's safe. This gene therapy, which is really a first for eye disease, but also a first in all of medicine, has already gone through all those trial and research steps. So now is really to identify the patients who are out there who have this mutation and who can now become the first patients to be treated here in Canada. The treatment is already has started in the US since end of 2017, and several patients actually from Montreal, from Canada, have already traveled to the US to receive the treatment. But now this has now come to us so that they can now receive the treatment within their own city, within their province, instead of having to to make these trips elsewhere and, outside the country to receive the care.
I think that, okay, I have so many questions here. I think it's interesting. I'm talking from the perspective of patients as well, right? So when I look at, when you're saying how this is really a first in all of medicine, or much of medicine, it's because the eye is accessible for doing this type of therapy, right? And we can visibly, examine the eye, post intervention to see what's going on, right? So in many ways, I don't want to say people with inherited retinal diseases are lucky per se, but when you're looking on this on a continuum of different types of inherited diseases, a lot of the ocular diseases will be more, I'm going to say, more readily addressed or earlier on in the implementation of gene therapy overall. But the flip side of that, from a patient's perspective, and this kind of leads me to my next question, is how am I supposed to, I don't want to say trust this, right? Like there's a couple of years of data behind this, and what happens if I want to just, I could qualify, and just to be transparent, I wouldn't qualify for this study. The gene that's mutated in my family is called RPGR, but and I know there's a lot of clinical trials ongoing for that. But if someone's saying, "Hey, I want to see more data on this. I want to wait until there's something more concrete. I'm still very early stage in the disease." Have you come across that resistance from patients? And if you have, what advice would you give to them? Would you say, and then it might come back to what we asked earlier about, this is a progressive disease, but I was hoping maybe you can just speak to that a little bit.
Yeah. And again, I think, like you said, Sean, this could lead to a multiple hour discussion because this is such an interesting topic. I think, from a safety and history point of view, like you said, this treatment, that the first one to come to Canada, Luxturna, or the one treating the RP65 gene mutation, we're hearing about it now, but this actually, in the scientific or ophthalmology community, this was over 25 years coming to get to this step. So yes, it's a new breakthrough, but if we look at the whole history of how this gene therapy came to be, from the initial identification of the RP65 gene back in the early 90s, to being able to isolate this gene mutation, to being able to create a gene therapy product. And in this case, we're very lucky because it's a very small gene. So it was able to, very nicely package it into the vectors that are then implanted, going through an animal model, the Breyer dog, that was able to benefit from the results of this research. Then moving on to the human studies, phase 1, 2, 3, and then 4 even of this step of the study. This was 25 years in the making. So this is new, but it's not so sudden that, it's not results that came through, within a couple of months of research. We have years of studies on, we have some patients that date back over 10 years now who've received the initial preliminary results. So we have data going back to show how they've tolerated, how the treatment and how they're doing now. So, and I think it's valid questions, for patients who were previously told, well, you have an untreatable disease. There's nothing to do. There's no need for follow-up. To suddenly be told, please come and please be seen. It is a big change in the culture and in the attitude. So I think it's important that patients either hear this from their doctors, people who know about the research, the whole process, the painstaking, very meticulous scientific process that goes through before getting each of these approved and coming to market, and so that they know also to, if they haven't seen an ophthalmologist for many years, that this is the time that things are changing and it's worthwhile to come back and get a genetic test done, because that wasn't necessarily done, in a standard routine, even maybe 5 or 10 years ago. And also this sets up the precedent that because it's gene targeted, I think it's advantageous in the sense that there's not going to be non-targeted treatments that come out all the time, that if people sign up for one treatment, they're not eligible for another one a few years down the road. If this is a treatment that's specifically targeted towards a specific mutation, to have gotten to this step, this is based on all these past decades of research. And I think what I see is more a rapid finding of key gene therapy solutions to different mutations, but based on this very robust decades of research that was used to develop the first gene mutation. So all the new ones will be permutations of this evolving technology that's taken time to establish itself.
Yeah, I think that's very interesting, right, for them to know that if they do the genetic testing and they happen not to have the mutation that can be treated right now, it doesn't mean that wasn't worth it, right? Like, I mean, at least they will know their mutation and they might have a treatment in the future.
Definitely. And I think this now is the time, right? Like you said, there hasn't been a very robust building of this genetic database in the past. And this is why, when we have this treatment now, we're hitting a bit of a wall, because maybe there are people who have the RP65 mutation, but who were never tested before, right? So we have a little bit of a lag in starting the treatment, because we have to now try to test everyone who was previously diagnosed but who didn't have this genetic mutation testing done 5 years ago. So imagine if we do these tests now and we get the results. Maybe they don't qualify for the current RP65 treatment, but at least we'll know. For example, you have RPGR mutations. Perhaps you have Stargardt mutation, and then when that gene therapy comes down the road in 2 or 5 years, they will already be ready, so that we don't, again, have this lead lag effect down the road when we get to that point. They will already have the information in hand, and that's really a powerful tool. And so, I think it's really, you're completely right. It's about changing the perception of what the genetic mutation test means, both for patients and for colleagues. I still have colleagues who see a patient who, for the first time, they tell the patient, well, I just diagnosed you with retinitis pigmentosa. Please come back in 5 years, because there's nothing to do. Like, even very recently, I've heard of patients coming to me with stories like that, so, and that's really not the way it should be anymore, right? The science is advancing, and every year there's change. Like, they shouldn't be coming back in 5 years. Things are changing every year, every month. And same thing for patients, right? I've had patients who in the past said, well, what's the purpose of doing genetic testing? You don't have any solutions for me. Like, it doesn't change anything in my day-to-day life now. But, if you take it into this broader perspective that it's opening horizons for future treatment, then, we really see the importance of doing these tests.
And talking about day-to-day life, right? I think, like, it's a question they might have is, like, I mean, what exactly does success feels like, right? So would you have any story of a patient that was treated already and how much their vision improved and how much their lives have changed, like, so people can have an idea what they can expect if the treatment works?
For sure. So, the one that's been approved, as I mentioned, Luxturna, that's the commercial name, or Voretigene and Pavrovac. As I was mentioning, there have been patients all over the world, mostly in the US, because they've started enrolling and treating since end of 2017, who've received the treatment. And most of these patients are actually children, because, the disease Leber's congenital amaurosis manifests itself since childhood. And as we mentioned, the earlier you treat before there's severe loss, the more likely you're going to gain back visual function. So, there are stories of children who have received the treatment, and within a couple of weeks, and this is from reliable information from the patient and from the treating physicians, where the children mentioned that all of a sudden, they're in primary school, the board looks different. They're perceiving a lot more in their environment than they used to do. And, very recently, there was also a story of, I think it was in the news, where a Montreal boy who went to Philadelphia to get treatment for this mutation as well, the Luxturna. And he's come back, he's had a successful healing process. And, when he was asked what he sees differently, he says, well, for the first time in his life, he can see the stars at night. So, for example, these are things that people who don't have visual deficits take for granted, but for these patients, especially these kids who have a whole lifetime of vision ahead of them, this makes a world of difference in terms of what they can do at school, with their friends, in their day-to-day family life, and what it means ultimately for their whole lifetime and their career. Because right now these treatments, with the data we have, we don't know exactly how long the effect will be, but we can assume for now that this one dose should at least ultimately cover many, many decades, or ultimately a whole lifetime. So getting one treatment by having the gene therapy supplement the missing or the mutated gene really makes an impact lifelong.
And even if the cases that do not see a remarkable improvement in vision, it could have prevented like the progression of the disease, right? So it doesn't mean that the treatment didn't work. Does that make sense?
Yeah, I think it's, again, everybody, it's really a case by case study. So everyone, responds to the treatment differently, and it's very dependent on, as we're saying, the degree of the disease, the age of the patient, what they've gone through before. Even in some studies, not for RP65 per se, but, for example, for gene therapy in choroideremia, which is another inherited retinal disease affecting mostly male patients because it's X-linked. You have brothers enrolling in the study, and both ultimately, having different outcomes. So it's really an individual experience in terms of the surgery and then the final effect. But ultimately, the goal is to make the patients better. But the level to which they improve will be variable depending on it's multifactorial. It's based on many factors surrounding that person. But the goal is obviously to give them back function and to help them keep their visual function for as long as possible without any severe declines over time.
Okay, so I have about 4 different directions I want to go here. I'm sorry, this is awesome for me. So a couple of things. So when you're talking about patients, they can have variable outcomes. I think it's important for people who are listening that you're also talking about an approved therapy with a very favorable risk profile. So the risk reward ratio here is quite favorable, right? So fairly low risk intervention with potentially life altering reward, right? So I think that that's a bet that I would place any day. I have as you're talking about some of the ophthalmologists still, telling people, hey, go home, come back in 5 years. It just reminds me of a story from the well, you're very familiar with the Argus 2. I think, were you not part of the first team to implant these in Canada?
Right. Sure.
Yeah.
Yes.
So the Argus 2, the bionic eye, so to speak, or the retinal prosthesis. I remember after that it was approved, reading something, and it would have been Robert Greenberg, who was the CEO of Second Sight, was talking about how they were running into trouble actually getting patients. And it's like people didn't, they thought there'd just be this flood of thousands and thousands of patients that want to get this treatment that's available. And there's a lot of people that just didn't even know about it, right? And I'm sure that there are many clinicians right now, as well as patients, that don't know about, a treatment for RP65. And, hopefully this podcast can be one vehicle by which we can help educate patients and clinicians, right?
Definitely.
You mentioned that the RPE65 is a small gene, and I'm assuming this is why it was one of the, based on prevalence and based on size of the gene, I'm assuming this is why it was one of the, or the first one to really be pushed through trials. Does that make sense?
Yeah.
So is the, and maybe you don't know the answer to this, I don't know, but because there are a number of different genes that can be affected and that can ultimately lead to or manifest as retinitis pigmentosa, is the size of the gene a barrier or the barrier to delivering this gene, I guess, using this viral vector or this packaging to shove the gene in and then stick it into cells? Is the size really important, or is that have basically been dealt with in general?
I think there are very ingenious ways now of dealing with that. So definitely, in earlier years, size of the gene product that you want to package is an issue. So in this case, like we said, the RP65 gene is quite small. It's around 62 kilodaltons, and it fits very nicely in the vector, which is one of the more popular ones used for gene therapy. It's an adeno-associated viral vector. Like we were saying, with COVID year, I think pushing through all this new data about vaccines, mRNA, is going to kind of help, and I think take away some of the fear or the stigma around having these viral gene vectors. For some other diseases where the gene mutation or the gene product is bigger, one example I can give is, for example, Usher syndrome. That's one of the forms of retinitis pigmentosa, where many people also have associated hearing loss. That's also one of the more common types of genetic mutations for RP. The gene product is much bigger, and hence it would not be able to fit on a standard adenovirus vector like the RP65 has been able to. So in the past, what they've done, or what actually current trials for that disease is doing, is that they've used other viral vectors. And in this case, they've used what's called a lentivirus viral vector, in which you're able to fit a much bigger gene product, up to, I think, 10 kilobase pairs, and sometimes what they've also tried is, if the gene product is too big, they've kind of tried to separate it into two pieces and have it delivered on two different vectors, So these are some of the earlier solutions that were used to bypass the issue of size. And then obviously, what's really exciting now also is the introduction and use of the newer CRISPR-Cas9 technology. I'm sure everyone has recently heard about, this technology being recognized for its value by two of its researchers winning the Nobel Prize just last year, Dr. Doudna and Dr. Charpentier. And so the idea here is that basically you are, instead of just delivering a whole gene product that's missing or malfunctioning in the original person's eye or body, you're delivering a gene product that's going to go try and repair the error that's at the primary site of the mutation. So the idea is, some people are calling these kind of DNA scissors, where you would try to snip out the faulty gene and then introduce the correct sequence, so that then the initial or the native gene is now going to be able to produce the correct gene product. So the fact that you're snipping out and then you're trying to introduce the correct gene sequence, this also takes away some of the idea of packaging a whole gene product and sending it in, and instead of just replacing, you're talking about correcting. So really, we're talking about new different technologies coming to the forefront. And once again, as Sean was mentioning, because the eye is such a special place, right? It's a very enclosed space. You're able to access it directly without having the gene product go through your whole body. You're able to very precisely deposit the gene product at the level of the retina where you want it to act. So CRISPR-Cas9 is also one of these technologies that has been taken up in ophthalmology and in retinal research. And there's actually a current study using that technology, studying its effect in treating a different form of Leber's congenital amaurosis, which is Leber's congenital amaurosis 10, which is caused by a different mutation, the CEP290 mutation. And this is also a study that's generating a lot of excitement because it's the first, once again, in ophthalmology, but also in the whole world of medicine, where you're once again directly administering the CRISPR-Cas9 technology to modify and to correct the mutated gene disease.
So I think that just a couple of things there. You mentioned, I think it's Jennifer Doudna, right? Who
Yes.
There's actually, if I'm not mistaken, there's a book by Walter Isaacson who authored, Ben Franklin's biography and Steve Jobs and a number of other people. I think he just put a book out talking about her. I could be wrong, but
yeah, I think so.
I thought I read that somewhere, and I think it's on my reading list somewhere, but I didn't get to it yet. So I just wanted to also talk to you about, there's so much excitement that's been happening in this field recently. What if you had a crystal ball? I guess if you crystal ball, you would know the answer, but if a hypothetical crystal ball and we're trying to peer 2, 3, 5, 10 years into the future, knowing what has happened in terms of developments in recent years for gene therapy for inherited retinal diseases, where do you envision that field, in, let's say 5 years from now, give or take, or where would you like to see the field? Obviously, you'd like to see the field, light years ahead, but where would you envision that the field could be? And to put it in context, like, do you think that there could be approved gene therapies for RP or other forms of inherited retinal disease, that span multiple different underlying mutations? What do you think?
I'd like to see the field evolve in a way that makes that treatment very accessible. Because right now it's in the earlier stages. We're talking about the concepts of what we're trying to do, and the principles hopefully make sense, when we break it down into these pieces. But, to make the whole operation work, there's a lot of logistics that goes into it. We're talking about choosing specific hospital centers that are able to, first of all, identify these patients correctly with the correct genetic tests, because genetic test interpretation is also something that's quite complex. Having the right, first of all, the surgical team, of course, that's what people think of. But, behind the scenes, there's a whole lot of logistics about how the gene therapy is delivered, how it's pre-processed, whether there's, access to easy specialized pharmacies that can store, that can prepare the gene product to be used in the right time frame. It's kind of like, like the COVID vaccines right now, right? Like 2 doses has to be at minus 80. You have to keep it at this temperature. So the product is quite fragile, and you need a whole tight schedule of logistics and people and equipment to make it work. Like, for example, for Luxturna, once the product is prepared, you have to use it within a 4-hour time span, right? Otherwise, it's no longer active, and it's very tightly controlled, and then you have to work with the anesthesia team, have the patient ready. Like, are you at the right surgical step to be able to administer it? Is the dosage right? Do you have the right needles, et cetera, et cetera? So what I hope to see is that, the science advances so much that this now becomes, like a routine treatment. Someone comes to your clinic, is diagnosed with RP. You can do a quick genetic test and say, this is your mutation, and we can schedule for treatment within this short time span, and not only limited to only, for example, now University of Montreal is one of 2 centers in Canada to administer it, the other being the University of Toronto, but that it would become something that ophthalmologists across the country have access to and can easily administer without having to think about the logistical difficulties of, obtaining, maintaining, and giving the treatment. And I think the other direction, like we said, is, RP is such a heterogeneous, it's a term that englobes so many diseases, and that's something that I think patients sometimes have a hard time grasping, right? They see RP, like in the news, there's a treatment for RP, and they think that it's right for them, but, it's not. But we hope that, with time, all the different mutations will be more easily identifiable, and that we will have a gene therapy for each and every single one of those mutations, be they a very common mutation, be they a very rare one, be they a very large gene product or a very small one, that they all become at the same level, easily reachable for all the patients.
Yeah, that's all very exciting. Cynthia, thank you very much for sharing all that. If there's one thing that this episode will do, I think, is to like encourage like patients that have retinitis pigmentosa to take action about their diseases after so many years that they've been told that there's nothing that could be done. So if you could put yourself like on the shoes of a patient, right, that it's now hearing about all that, what are the steps that they're going to have to do until they actually get tested?
Right. So many of these patients, some have, once they're diagnosed, receive information, receive news from one of the vision rehabilitation centers they're affiliated with, be it, in the province of Quebec, you have Nazareth, you have the Mackay Lethbridge Center, you also have other centers like Le Bouclier in the different regions that people live in. So I would say it would be, if there's a regular contact with one of these centers, reach out to those centers first. We don't have to reinvent the wheel to try to get back into the system. If there's already a person that's your link or contact, go to that person. The other thing I encourage people to do as well is, if they're not familiar with the Fighting Blindness Canada webpage, is to also get information from that webpage. I'm part also of the committee for Fighting Blindness Canada, where we're trying to also create a Canada-wide registry so that all people who have had previously been diagnosed with inherited retinal diseases, be it with a genetic testing or not, is within the registry, so that we can easily have access to communicate with them and contact them should there be a new treatment, a new study that's coming out, so that everyone knows, where they're at, both from the patient side of things, of what's coming down the pipeline, and from the physician side of things about how to reach the patients. And also, I do see a lot of my own patients in my practice, but I'm also happy to get, consultations from my colleagues who have some of these patients already in their practice, and they'd like to have them seen for an accelerated genetic testing process. And right now we're working with for a limited time, we're working with Novartis, who are the distributors of the medication Luxturna, in accelerating the turnaround time for getting genetic testing done and having the results back. So I would say, it's, if you hear this podcast or if you hear about these, exciting news, it's the time to sort of, you can reach out to us, and, we're more than happy to kind of go through the process of explaining what the steps are, and of doing the tests to see, what your mutation is, if it hasn't been tested yet, and whether you're a candidate for this approved treatment.
So Cynthia, on that note then, so if I'm a patient listening to this, which I am, but do I go to my ophthalmologist and say, hey, I heard this podcast about this treatment and maybe I could qualify? Mm-hmm. Do I do that and have the ophthalmologist reach out to, the department at the University of Montreal, or what would be the process to get that initial contact for a patient?
Yeah, so that's perfectly, if you, that for sure, we know each other, so that would be a easy, kind of referral, but that sounds like a perfectly good pathway. We're also actually looking to see, right now I'm ordering a lot of the genetic testing. We're also trying to see if different retina specialists and other ophthalmologists can actually also try to order the test first. Because even with the accelerated process, it's a couple of weeks of turnover, so that, we're trying to see if there are ways we can get the testing started and, have the ball rolling, so that, once we hit the ground and we get to the next step of doing more sophisticated imaging tests, that the genetic testing results are already ready, available, and back for us to kind of review together. So So yeah, so that could be discussed, either as a referral to the University of Montreal, or if their treating retinal physician is able to order the genetic testing and get things started, that would be another pathway as well. We also work very closely with the Institut Nazareth-Libride, the INLB, as I had mentioned, as well as the Mackay Center. So patients who are affiliated with those centers can also reach out, and they have ways of reaching into our system to set up appointments and consultations.
So sorry, I'm gonna keep diving on this just for a minute. I probably have 3 or 4 family members that I can definitely refer, because I underwent genetic testing at the National Eye Institute back in 2009. So I know the mutation, but in talking to Dr. Flavio Resende in our last episode, he emphasized how important it was for patients even within the same family. They think they know the mutation, they still have to get tested. So I can probably serve up a few people very quickly. But when someone gets genetic testing, just for the audience to understand, is it just a blood test, or what is involved for the patient? Like a genetic testing can sound kind of scary if you don't know what to expect, right?
Yeah, so the classic and probably still the gold standard is just a blood test. Yeah, it sounds scary, but it's no different than having your blood drawn to check, to check whether your blood sugars are good, or, just like a routine checkup where you get a blood test. So it's the same thing. And we don't take too many. So so it's not, you're not getting like 10 tubes of blood tests done. Alternatively, there are other tests. There are some other tests that are commercially available now, where it's simply getting some genetic material from your saliva, So actually the tests, some of the ones we're sending out now, we just need a saliva sample to then be sent for testing. The saliva samples are more ideal for adults because we do need a good volume, and sometimes for kids it's actually hard for them to to spit in the tube and to get the volume that we need. But both are equally effective, in determining what the genetic mutation is. The advantage of the blood sample is that most likely if you send it off for testing, you can keep a portion of it for example, in some cases, genetic testing comes back and it's, non-conclusive or it's negative. That doesn't necessarily mean that there's no mutation. It just means that perhaps there's a gene at work that we're not familiar or that has not been identified yet. So being able to keep some of that blood sample allows you to retest it at a later time should there be more information about genetic testing. And right now a lot of the tests are what we call panel testing, where you have a panel of tests. You're testing for 20 genes or 50 genes, but the science of the genetic testing is also giving us more opportunities, and it's moving towards what we call whole genome testing, where instead of just testing certain genes, the technology allows us to test for many, not just the genes that we know, but for to look more broadly as well. So that's an advantage, where you can do one sample but that could be used over time for a long time or for many years.
I think that's it's interesting. I think the database you're building of patients is genius, because then patients they're in the system. Now their mutation is known, and basically it's like, hey, if there's something that could potentially help me, contact me, right? Rather than trying to follow up and interpret, hey, could this help me, and whatnot, right? So that's great. Yeah. I think we've been running a lot. Bruno, do you have any other questions you want to throw at Cynthia for this round one?
That's for me today, but we would definitely want to have you again. There's so many questions that I would like to ask, but I think today we had enough of your time.
Yeah. No, it was a great pleasure talking, and there was, it just felt like a discussion amongst friends. So that went by really quickly, and I really appreciate the opportunity to talk to both of you and share some of my experiences.
Great, and then hopefully once COVID has become a thing of the past, maybe we can actually sit down as friends and have a coffee and talk more about retina. So until that time, Cynthia, thanks so much for joining us. It's truly been a pleasure, and we're definitely going to make sure as people listen to this, I think that we're going to be flooding the phone lines at the university. But no, thanks seriously for sharing your experience and knowledge. It's truly a pleasure. Sorry.
Same here. Take care,
all right,
guys.
Take care. Bye now.
Thank you, Cynthia.
And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.